Thanks, everyone. It's so lovely to see so many faces in person. So I'm going to talk about our strategy for health outcome phenotyping, which is a huge topic, but I'll do my best in 10 minutes. So UK Biobank's health outcome strategy over the next few years can really be summarised in four main different strands. So the first, which has been well underway for several years now, is cohort-wide linkage to the key electronic healthcare records that exist across this country, so national death and cancer registries, hospitalisations, and where we can, primary care data, and I'll talk a bit more about the issues with regard to primary care in a moment. The second strand to the strategy is, in the future, we'd really like to be able to link to specific data sets to obtain more granular information on particular health outcomes. So this could be clinical audits and registers, histopathology slides for cancers, imaging scans for stroke and dementia, and so on. The third strand is to collect new data on our participants in order to obtain more data on health outcomes. So this could be through web-based questionnaires, it could be through apps, as discussed in the last session, to obtain more granular detail about prodromal symptoms, changes in behaviour and so on, or bringing participants back to specific assessment centres to obtain more detail about their health outcomes. And then the final thread in all of this is, once we have all of these data integrated into the resource, is what analytical approaches can we use that can combine all of these data across these diverse sources to better define health outcomes for researchers to use. So these are the current linkages that we have already in the UK Biobank resource. And what you can see here of note is that for primary care, we only have data on just under half of the cohort up to about 2017. And this is because at that time, there was a new requirement that was introduced that meant that GP practices essentially had to opt in to approve the release of primary care data from the system suppliers to UK Biobank. And we spent several years doing extensive piloting, and no matter what we did, only about 20% of GP practices would actually approve the release of the extraction of those data. However, at the start of the pandemic in 2020, emergency legislation was introduced that enabled primary care data to be released automatically from the GP system suppliers in England to UK Biobank, specifically for the purposes of COVID-19 research. And what that meant was that, crucially, it was no longer the responsibility of GPs to approve the release of the data. The data just automatically flowed in accordance with the participants' consent. And these data for England, the primary care data, enabled really vital research to be performed, particularly into the clinical determinants of severe COVID-19. So that emergency legislation ended in June of this year, and those data are now no longer available for COVID-19 research. And I think what the release of these data has really showed us is actually what has been lost in us not being able to access primary care, not just for COVID-19, but for heart disease and cancer, arthritis, and all the other health conditions that are of equal importance. So we are currently under discussions with the BMA and the Royal College of General Practitioners to find a mechanism through which primary care data can flow automatically from the system suppliers to UK Biobank for those consented participants to fulfil those wishes. So those discussions are still underway. So why are we so bent on being able to characterise health outcomes accurately? So obviously it's important to identify as many events of a disease as we can in order to maximise numbers and to ensure there's representativeness across the disease spectrum. So we want to find out who has mild and severe disease, who has different subtypes of disease in order to look at different etiology. Identifying as many cases as possible increases the power for researchers to look at associations between risk factors and disease outcomes. And if we can reduce the number of false positive outcomes, that is really what we're aiming to do, is that has the largest impact on study power. And of course, we also want to increase the biological specificity of disease outcomes to enable researchers to better understand the determinants of different subtypes of disease, which we know for many instances has a different etiology. So how are we going to go about doing this? Well, what we want to do is to link to a wide range of healthcare records and other information to ascertain as many events of a wide range of diseases as possible. And from there, we want to be able to generate research-ready or off-the-shelf health outcomes across all of these diverse healthcare records for researchers to use. So over the next few years, our intention is to link to a whole new range of specific healthcare data sets that contain more granular information on health outcomes. So the ones shown here are those that we're actively pursuing at the moment, and these have to be assessed based on the scientific added value over and above the existing data we have, the quality, comprehensiveness of the data, the cost, and the feasibility of actually doing the linkage. And Anthony will talk a bit more in this session about the proposed linkage work to the eye and vision data sets. And we're currently scoping out new data processing pipelines that will be needed in order to more efficiently and quickly process, upload, check and integrate all of these new linkages into the UK Biobank resource. It's quite a big endeavour, and at the moment we need to streamline our current underlying processes in order to do this effectively. Specifically, we'd like to focus on being able to identify cancer subtypes, and there are three main strands of activity that we would like to focus on in order to do this. So the first is relatively straightforward, which is for us to link to enhanced cancer registry data. So this is data collected by the cancer registries that contain granular information on stage and grade to assess disease aggressiveness, prognostic indicators, and also treatment as well. We're also about to start some pilot work to assess the feasibility of linking to digitized histopathology slides, which will enable researchers to identify anatomical subtypes of cancer based on the actual cell structure. And Kezia will talk a bit more about the rationale for that later on in this session. And we're also exploring the feasibility of whether we can link UK Biobank participants with cancer to their tumour sequencing data where it exists, in order for researchers to better identify subtypes of cancer based on their precise mutational signatures. Now, we've talked quite a bit in the last session about the importance of research into dementia, but what we'd really like to do in the next few years is to get a better handle on how to identify dementia subtypes. Even if we were able to access primary care data for everybody, only about half of dementia cases that are coded in primary care have a dementia subtype. So we need to think carefully about what extra data do we need to collect to enable research into Alzheimer's disease versus vascular dementia versus all the other subtypes. So in talking with the dementia experts, many of whom were speaking in the previous session, there's a real appetite to potentially invite participants recently diagnosed with dementia, perhaps back to an assessment centre, because here we'll be able to collect a biological sample, we'll be able to form blood-based neurodegenerative markers, both performed at baseline and then also at the diagnosis of the disease, so you look at changes in time. There's a potential that we could perhaps do imaging scans on these participants so we can understand the pathological basis of the subtypes of disease. And of course, if you have participants face-to-face at an assessment centre, you can then assess the clinical presentation, cognitive function tests, including possible use of devices and other apps and so on, in order to understand diagnostic presentation and also progression of the disease. We're also looking to expand our online questionnaire in order to obtain self-reported data on specific health outcomes that are particularly difficult to assess through linkage to routine records. So these are the ones that are currently in development that we plan to administer over the next 12 to 18 months. So there's a pilot study has just gone out this week. Actually, we're just starting a sleep disorders questionnaire, followed by one on neurodevelopmental disorders, then a repeat of cognitive function, and then one that's focusing on particularly rare neurological disorders as well. And all of these conditions are pretty difficult to ascertain through linkage. Once we have all of these extra data incorporated into the resource, we then need to generate research-ready health outcomes. Currently, there are no standards for selecting and combining different codes across the healthcare records, and there's a surprising variability in how researchers define the same disease. So we need an approach that can combine all these different data to generate outcomes that are valid, comprehensive, and scalable, and so on. So we think we're going to plan to, there's three main avenues that we want to try and pursue over the next few years in order to generate these research outcomes. So the first is approaches underway where we're mapping all of the coded data across the healthcare records, plus the new ones that are coming online, into a common coding standard. And this will be a 4-digit ICD-10 code. So it will be a natural extension to the first occurrences field that already exists in the UK Biobank resource. And this will hopefully obviate the need for researchers to actually integrate the raw, complex electronic medical record data, because these health outcomes would have been derived from those codes. A second approach is to assess the feasibility of whether we can actually integrate into UK Biobank some open source tools and algorithms that already exist that define health outcomes. So the obvious one here is to collaborate with HDR UK Phenotype Library, where there are hundreds of clinically based algorithms that combine all these different codes, and can we incorporate that into UK Biobank to save duplication of effort from researchers having to do that work themselves. And finally, over the longer term, we'd really like to be able to support data challenges to enable researchers to explore machine learning type approaches that can classify health outcomes based on all of the existing data included in the resource, not just restricted to code-based algorithms. So all in all, I mean, it's an ambitious programme of work. We've got a lot to do over the next couple of years. But if you have any ideas about current linkages that you think would be of important for UK Biobank or important for your research, or any other approaches that you think is worth talking to us about how better to define health outcomes, please do get in touch, and we'd love to hear from you. So thank you very much.